Chapter 16, Theory Video 3. Now we are going to discuss about the transcription factors in eukaryotes. Now we discussed and we learned that the prokaryotes have operones. But eukaryotes don't have operones. Instead of operones, genes are expressed and they are controlled by transcription factors. So this is a transcription factor is a protein that binds to DNA and it will affect in a manner whether the gene is transcribed or not. So it is what is going to it is what is going to inhibit or allow. Either the transcription factor will it will allow or disallow, but at the same time it will make sure that the genes are expressed in the correct cell at the correct time and the correct extent. So this is one of the things that you need to remember and understand. Now correct cell at the correct time. So this time, and there is a reason how all of these our biological systems are connected to each other. They are interdependent, and there is a relationship between times. Now, for example, the hormones, the enzymes, the proteins that are all produced during daytime are not produced at night time. At the same time, when one activity is taking place, then corresponding genes need to be expressed. So these are controlled by these transcription factors. Then they say that in humans about 10% of the genes are there to code for proteins that act as transcription factors. Then these proteins or transcription factors get attached to promoter region of a gene. So that allows or prevents the transcription. So this transcription, the presence of a transcription factor will either decrease or increase the transcription of a gene. So if it is to be expressed, then the transcription is increased, and if it is not expressed, then it is reduced. Then there are many different types of these proteins known as transcription factors, and they have different effects. We are still studying about these things. Not a whole lot of transcription factors have been found. So only a very little knowledge is there for the moment. If you if you want to now when we have to understand what a transcription factor does, we need to actually go into the relationship. We can actually compare the prokaryote relationship with the operon. So something similar to that, but in a different way, takes place in eukaryotes. So there are different types of transcription factors, like I said. So there are general transcription factors. They are necessary for this transcription to occur. So that they produce a protein complex and attach to the promoter region to allow for the transcription to occur. Then there are other transcription factors that allow different genes to act in a proper order, that is, in a correct sequence. So when a zygote develops, the parts of the body should develop in a proportionate manner. If some parts grow larger than the others, it is going to be a problem. then it is a transcription factor that is going to be responsible for the determination of sex in mammals. Then there are transcription factors that respond to environmental stimuli. When there are high environment temperatures, when there is less light, how people become sleepy and all of those things, like hormones, testosterone, and their the effect of these hormones are controlled by transcription factors. Then the products of proto-oncogenes, which is something that you must have discussed by now, and tumor suppressor genes, tumor suppressor genes, and these are required for the control and maintenance of the body. They regulate the cell cycle, growth, and apoptosis. That is the natural cell death, programmed cell death after it has been used out. So these are the factors that are going to these are the transcription factors, or the proteins that are responsible. These are proteins which are responsible for certain activities inside the eukaryotes, and these transcription factors are in place for the operons that control the same thing in prokaryotes. Transcription factors are quite similar in plants and animals. So we discuss here the gibberellin control, the gibberellin, how it controls seeds germination in wheat and barley by stimulating the synthesis of amylase. So this is a situation where a hormone influences transcription. If we apply gibberellin to barley, as a result, mRNA coding for amylase is increased. Then, as a result, what happens is gibberellin breaks down DELA proteins, DELLA. So this molecule, DELA protein, is a repressor protein. What it does is it inhibits the binding of transcription factor. For example, phytochrome interacting protein, PIF, which is going to attach itself to the promoter, like we discussed earlier. So this DELA protein is broken down, and as a result, PIF will attach to the promoter. As a result, transcription can take place, and the amylase production will continue. Right. So that is how the gibberellin is able to act within these cells and improve the increase the rate of the seed germination. Write down a note. Eukaryotes, transcription factors in eukaryotes is the topic. In eukaryotes, instead of operons found in prokaryotes, there are transcription factors. It is a protein that attaches to the DNA within brackets, usually to the promoter and either allows or prevents transcription from taking place. There are many transcription factors with different functions or effects, such as general transcription factors which allow transcription to occur, comma, allowing genes to be activated in correct sequence, comma, determination of sex in mammals, comma, allowing for responses to responses to some environmental stimuli, comma, regulating the cell cycle and expression of effect of some hormones like testosterone. Leave a line and write. In plants and animals, transcription factors affect in the same manner. In the following example, gibberellin which controls seed germination if applied to barley seeds will result in the increase in transcription of mRNA coding for amylase. This is done by gibberellin, which causes an oppressor molecule, a repressor protein called capital DELA protein to break down. This allows A transcription factor such as phytochrome interacting protein PIF to attach to the gene promoter and transcription to proceed. I hope you understood. Please draw these 16.14. If you don't understand, ask me questions. If you have understood everything, I would suggest you to do the end of chapter questions.